Good afternoon. I'm Dan Freed, Director of the Harvard Center of Mathematical Sciences and Applications, or CMSA. So it's a pleasure to welcome you here to our annual YIP lecture. This is one of several lectures that we do at the center that are public lectures each year. You can find previous YIP lectures and other lectures on the CMSA YouTube channel. And I encourage you to check out our website and learn more about the center. Today, we're very pleased to have Regina Barzilay, who works at MIT. She works at the interface of artificial intelligence and health. And today she's going to tell us about machine language methods for designing drugs.
Thank you. Thank you very much for having me here. And, when I was deciding, what to talk about, I thought I have no clue who is in the audience, correct? Maybe many of you know better chemistry and biology than I do. There are other people who know math better than I do. So it's like kind of average distribution of the talks. But I was just thinking what I would want, what I would have wanted to learn about this field, 6 years when I was 8 years when I started working on it. It's, we at MIT started working on small molecules maybe 8 years ago. At a time, ML for drug discovery was really, really small field. And through this process, starting from small molecules to proteins and to modeling cells, we kind of went through the whole trajectory. And there are still a lot of things that I'm learning. So this is not going to be a glorious talk to say we can solve all the problems and this is a problem and see how wonderfully AI solves this problem. It's not going to happen. What I'm going to tell is kind of tell you this where we made progress, but I will highlight in the talk, and to some degree it's going to be a talk of failure, some areas where we either don't have solution or we have a very bad solution. And my goal is not just to say, we're in terrible space. There are a lot of exciting things happening. But at the same time, hopefully, when I'm highlighting to you, places where the field is not really delivering, it will give you some new ideas where to step up in the field. So, 8 years ago, when we started working in the field, I barely knew what's going on. And I can tell that many parts of the space, there is a lot of science. And if you want to get into them, you need to learn a lot before you can contribute. On the other hand, there are like areas of desert where there is either no method or methods that don't work. So it's an opportunity for people kind of to step up and find the problem of their interest. And because we are like a small, cute audience, feel free just, to interrupt and ask me questions during the talk. I hope it's okay with my host. It's so that we are kind of aligned. And I will also ask you some questions. So it's a bit more interactive. Okay, so I just put 2 slides to kind of summarize how we're doing drugs today. And I selected a very big success story. This is a local success story. It happened in Boston. This is a drug that was developed by Vertex against cystic fibrosis. So for those of you who don't know, cystic fibrosis is a terrible disease which results in the fact that your body cannot properly kind of produce mucus, and it creates this, bacterial infection in your gut and in your lungs and all over, and the survival without treatment. Okay, so what I'm trying to say that it's really a bad disease, no solution. So what Vertex did, a decade ago, so they produced a new drug which totally changed the lifespan of people who have this disease. Now you can live with this disease in 50s, very controlled symptoms. The drug is extremely expensive. It costs around $300,000. And the reason it's so expensive, $300,000 per year, because it took over 20 years to Vertex to develop this drug. It was really, really a lot of science that went into it. And the key realization to how to address the problem of this disease was identifying the protein that was involved in creating the symptoms. So they identify in this protein, this thing doesn't work. So what they identified, there is a particular mutation in this protein that creates dysfunction, that it actually changes the way your cells produce mucus and creates a whole slew of diseases. So once they identify this particular space for mutation, then they came in and identify a binder to change the way this protein is functioning. So the point is, what was key to the solution? First is to identify how, when you fold this protein, if you have mutation, how it stopped regulating correctly, this mucus production. And the second piece is to make a fix and to make it work correctly. So in this case, this was a successful example of making it work. It just took 20 years. So the question is, can we make it work without spending 20 years? And again, today we have lots and lots of diseases for which we don't have any reasonable treatment. So roughly speaking, if you're looking at the whole landscape of methods that exist in machine learning for drug discovery, I would roughly divide them into 3 buckets. The first bucket is understanding the disease, and understanding the disease biology on the molecular level that you know what to fix. So in this case, they identified a protein that was a culprit. The second part, while you understand what you need to fix, is actually to create a chemical that can do the fixing. And the third piece is a piece that I'm not going to be talking about, is clinical trials and how to organize it. It's a very big area where billions of dollars are spent, but we're not going to be talking about it. We're going to be talking today primarily in this first stage how to understand biology and how to create the chemistry. Now, I should say ahead that most of the successful application of machine learning in discovery happens in the second category. It's really molecular science. We typically assume that we either know the target or we have an assay that help us to identify what needs to be fixed, and most of the work is actually how to fix it. So when we are thinking about, how we are fixing these proteins and how we are creating the drugs, there are typically 2 big directions within drug discovery. Both benefit greatly from machine learning. The first one is phenotypical discovery. So what it means is that you have some assay that can measure that if you edit the drug, did the right reaction happen? The closest one would be maybe in cancer cell lines. There is some assay that can see whether you kill it or not, if you kill the cell. So this is a phenotypical thing. You do not necessarily talk about the mechanism, how it happened, you just see the outcome. The very different type of discovery is target-based, where you really understand what happens, how the whole mechanism of killing happens. And in this case, you truly rely on biology to identify the right target and the way you want to modify it. So just to illustrate you the difference again, for instance, if you're thinking about antibiotic discoveries. In case of antibiotic discovery, there are lots and lots of PhD theses written in Harvard, MIT, and other good places, which try to understand what is the best way to kill bacteria. And there are multiple targets which are identified, which again are studied, that if you can modify their behavior, then you are likely to get an antibiotic. So this is an example of one of these targets. Totally different approach is to say, I don't really understand. I'm not going to be speculating that I have to go after this target. I would have an assay that can screen my molecule and says that if the killing actually happened or it didn't happen. So in this case, you can assume that you have a very big set of molecules out there, assuming that you have, the right way to screen them. And typically, in industry, you can screen, few millions. This is a set of a typical library, and you can find your molecule, and then you can start modifying. So I just want to show you one successful example. That's how we actually, for the first time, identify a useful molecule. It was a work with Jonathan Stokes, who was a postdoc at the time at MIT. Now he's a professor at McMaster University. And the way he proposed to address this problem was the following. He said, let's just take a collection of molecules. And we took a collection from Broad Institute. It's around 2,500 molecules. And then he took, the cells contaminated with E. coli. And he just saw which one of these molecules actually killed E. coli or didn't kill E. coli. Now, for those people who actually are familiar with machine learning, you can say 2,500 is a tiny data set. That's why I was really skeptical. But apparently, it was sufficient for us to do learning. And through this process, we actually were quite successful. I should give all the tribute to Jonathan Stokes, who thought about all the details. We just did the part in the middle, the learning part, but he did all the rest of it. We found this drug, which is called halicin. It has its own Wikipedia page. And what's special about this drug is that it works against resistant strain of bacteria. And it not only works on E. coli, but several others like Bowmania and C. difficile. Another interesting thing about this halicin is that it actually has a new mechanism of action. And this is an advantage of using phenotypical discovery, because this algorithm is pretty kind of unaware of the right biology. What it does, it just looks which combination of molecule kill the bacteria and which didn't. And it didn't say exactly how did you do the killing. And here I can just show you some few other examples of what this method delivers before I explain you the method. So in this case, this is a new work that Jonathan published recently. He was interested to find a molecule that kills only the organism of interest but leaves everything else intact. Because one of the big problems with current antibiotics is that they are like kind of bombs. They eliminate everything, including your microbiome and other things. So in this case, you can explicitly say, this species I want to kill, this species I want to leave. So you can do it. And many, many other things. You can really redefine what is your antibiotic. So now let's start talking about learning, how we actually do the learning. The reason you can do the learning, that if any of you are from chemistry, you know that there are particular functional group in the molecule that drive its activity. So for instance, you can look at these groups that I put on the slide and say those groups are known to be toxic, or those groups are known to facilitate antibacterial activity. Clearly, we don't know all of them, and that's why we need to do antibiotic discovery. So what we can do, we can model this problem as a very simple learning problem. What we will do, and the reason we kind of adopted this approach, because at the time I barely, I remember chemistry from undergraduate, so we were really not sophisticated chemistry or biology-wise. So this is kind of a computer science approach. So what you do, you take your molecules, you still remember, you can write them like 2D graphs, correct? You have your molecules in the training data. For each one of them, you're going to be recording their, their killing activity. There is a score which is called IC50, doesn't matter, some number that tells you how much killing this compound does. So you measure this quantity experimentally. This is what Jonathan did. And at the end, what you're going to get a data set where you have a graph and you have a number. And now what machine learning needs to do is to learn the mapping from the graph structure to a number. Does it make sense? So people in the older times, it's obviously not a new idea to do it. In the older times, what people have done, they would just take this graph, translate it into a vector, record different functional group or whatever, create some features, and then use, any machine learning algorithm to see which substructures contribute the most to the outcomes. Now, the problem is that if you're looking at a property which is complex, you may not even know what kind of subgroups really matter. So you want to learn it. You don't want to use fixed representation. You want to learn representation. And in this case, you can learn it, for instance, with graph convolutions. So once you learn the representation, I will just give you a hint how it is done. Once you learn the representation, what will happen? You're going to take these 2D graphs and embed them as vectors in some, high-dimensional space where a molecule which will have similar property are going to be close within that space. And again, you are going to be learning this representation specifically to a property of interest. In this case, for instance, we're doing for antibacterial activity. So again, how you can do it, you start encoding every atom in your molecule, in your graph, you encode it with some chemical features like the degree, the type of atom, and so on. So you record it based whatever. You start with some set of questions. I think we use like 16 features. And then what you're going to be doing, you're going to start doing this local aggregation through the convolutions. You're going to take your own representation and representation of your neighboring atoms and learn how to combine them to create a new representation for yourself. So what will happen through this process? First, you are kind of looking at yourself, and one hop features of the neighbors. And since we all are going to be updating at the same time, the next time you're going to have propagating features from a bigger neighborhood, and eventually you're going to summarize the molecule through the bigger neighborhood. So what we are going to get here is, feature representation for each one of the nodes. We can just sum them up and they will give us representation for molecule. Does it make sense? So I simplify. There are a lot of details you can actually do. The convolution also include the edges and other things. But this is the key idea. You start from simple representation for nodes and edges, and then through this aggregation process, you learn how to combine it. The objective that you're optimizing, it's your antibacterial activity or whatever other property you have in mind. Okay? So any questions so far? So now I will tell you, so the tool that we created, I think we published it in 17 or 18, it's called ChemProp. We try it on a wide range of properties. So you can see here, there is toxicity.
When you have these representations, do you sum them up or do you concatenate them?
So in this case, we just sum them up. So you actually learn, you do the embeddings on all the, for instance, atoms. And then at the end, when you finish the learning, you just sum them up. Yeah. And what happens here is that you can apply this model. I mean, there are variations of it. This is a paper, you can see the details. It's a very simple model. There are all these not important details, how exactly you parameterize, but key idea is very simple. You just parameterize message passing. Now, what you also can see here is that, this paper, and the tool is called ChemProp. It's very broadly used, maybe one of the most commonly used tools that we produce. You can see that you can apply it to wide range of property, to solubility, to toxicity, to bioactivity. You just need to have a training data where you have your molecule with a property and then it lands it. It's agnostic to the property itself, as minimal chemistry knowledge as possible. Now, this sounds fine. Now we're going to get to the part which sounds less fine. So I remember that this is an old slide. I did some keynote in ICML a few years back. And at the time, I wanted to see, because, as you know, in machine learning, the time moves very fast. Whatever you created in 2018 sounds totally archaic. And, today we have much better and faster and amazing methods. So I would be assuming that using this faster method is totally going to beat this ChemProp. So, I asked 2 of my students to actually take this ChemProp, which I thought was antique, and try it with all other different tools, make the comparison. And what I discovered, that barely there was any improvement. And I was really puzzled. I can tell you, and I will show you the numbers, that even if you are, it's a data set which is called TOX21, it's one of these relatively clean data sets. It's reasonably large. So I was really surprised why the model doesn't learn. And not doesn't learn, why all this new advancement, in graph representation and in modeling, did it improve these numbers? I was puzzled. And now, people try a variety of things. So I kind of left this field for a while, but I was once in a while looking at it and saying, what are the major things that happens and how we can make this number? Because you can say, is 80% okay? Yes, 80% is okay, but it's not fantastic, especially when you're looking at toxicity. You really want to move it much further. So the question was, can we use these big advancements that change natural language processing, that change computer vision, which is called foundation model? So again, what is the idea in the foundation models? That we can take these representations and learn it from some unsupervised data. So this slide, I thought I'm going to carry with me for the long time. This slide I did in 2019, before pandemic, until somebody proves me wrong. So the idea here is that foundation model that works in all other areas do very miserable job in small molecules. So let me show it to you what I mean by this. Again, what we want to do, I already explained to you, is that we can take the molecule, we can take the graph and learn how to summarize it as a vector and get a number. This process of summarizing, of learning representation, is done using some supervised data, correct? You can have like toxicity data. The exciting idea is that maybe I can take some very large raw data that is available and pre-train it. So I am going to be doing some other auxiliary tasks that would help me learn, and then I'm going to take this representation and apply it to my task. So here again, for people who didn't see, this is like a baby example. It's really old stuff. So in natural language processing, to get this representation learning, you can just take a sentence, remove the words from the sentence, and train the model to put the words back. So through this process, you're going to be learning how to, you're just getting a lot of free training data. So what will happen whenever you are learning this representation, you use the auxiliary task to learn how to map the words or the sentences into vectors. And you can do it pretty robustly because you can create as much training data with auxiliary task as you wish. And then whenever you start learning it on your task, you actually did a lot of learning prior to it. You can utilize your representation learning in a unsupervised fashion. Okay, so people try to do it. You know, and in chemistry, people always kind of go a few steps behind natural language. At some point, people said, oh, this is a great idea. It worked wonderfully in language. Let me just take it and apply it to molecules. And this is a paper from Stanford. They did exactly the same thing on graphs as NLP people did on sequences. They just took, you know, the graph and you remove some atoms and you try to predict them back. So this is your auxiliary task. Or for instance, you can just remove some functional group and try to predict it back. So you do the masking task. That's exactly what I show you on the case of NLP, where people were removing the words. Here we're just removing either atoms or some portion of the structure and try to predict them back, getting a representation. Okay, didn't work very well. But as I told you, every year I am committed to go and check what are the other models available for foundation model. And, you know, people do a variety of things. So what you can see, the first model, it's a contrastive learning model. So what this model does, it's kind of, again, the idea was very popular. It started in computer vision. You can say that when I'm learning my representation, it should be invariant to small modification, like say in the image. So if I take an image of the dog and it's slightly rotated, or I add shading, or I do something else, the representation of the dog should be very similar. So that's the idea. So you can do it with molecules. You can just, again, remove pieces, move the pieces, and you want to ensure that when you're learning representation, the vectors are very close to each other. Then there is another paper that comes from MIT that kind of claims, not claims, what it does a pre-training tasks. It tries to predict fingerprint. Fingerprint is a kind of a chemical summary of the molecule. And there are many of them going on. Some of them are more creative, some of them are less creative. But this is the funny part. And they actually, you can say, oh, maybe these models don't learn because they don't have enough data. Because when we are trained, you know, this foundation model in NLP, we really have huge data sets. Of course, we don't have the same huge data sets in chemistry, but still it's reasonable, you know. For instance, zinc data set, which have lots and lots of molecules, it's 1 billion molecule. These models are actually using 1 billion molecule. You have PubChem, which have, you know, over 100 million reactions. So there is quite a bit of data. It's not like totally sparse. And now you can see the numbers. So what I'm showing to you, let me explain what these things look like. So we took very standard data set. Everybody uses them. There are 12 data sets. And we ask for each one of these recent methods on how many of these how many data set, let's say the methods called MolClear, does better than ChemProbe. So there are 12 data sets. So you can see the MolClear does better on 3 out of 12 data sets. The closest is an MIT one, Chameleon. This one, on half of the data set, it works better. On the other half, ChemProbe does better. And if you look at the numbers below, the difference are really tiny. It's not really significant boost. So the story is that for some unknown to us reason, the foundation model that do amazing successes in proteins, they do amazing successes in language, they really don't work on small molecules. And it is a problem because the data sets for small molecule are actually small. That's why you theoretically can be really benefiting from doing some pre-training task or doing foundation model. So now I want to stop for a second and ask you a question. I have my own hypothesis, and it's just a speculation. I don't have any proof, but this is just a speculation. So if there is anybody in the audience who would feel comfortable to speculate or whatever, to produce a hypothesis, why do you think it is so that we don't see any value in small molecules for foundation models? Anybody? Yes.
Well, the data generating process is much more random, right?
Yeah, so this is what I'm thinking. And again, this is just hypothesis, and I have no clue how to prove that. Oh, sorry, I need to repeat. I am reminded we're at Harvard. The hypothesis was that it has to do with the data generation process. And this is what, again, similar hypothesis, because if you are looking at the space on which language is generated, those are valid English or whatever sentences which have meaning, that these words didn't just come together following some strict grammar, they actually encode some semantic meaning. If you're looking at protein sequences, again, those are evolutionary evolved sequences. When you are looking at these big spaces of small molecules, those are just synthetically generated molecules which are kind of kosher according to molecular grammar, but most of them never been even synthesized. So it's really not clear what are we learning from small molecules. But I don't know how to prove it. And I don't know if you're going to, let's say somebody asked me, but what happens if you take all the natural material, small molecule, would it change it? So I don't know. But this is a space, and the funniest part is that every year, literally every year, Czech, Clear, and NeurIPS, there is a new paper which does F-TAP evaluation and publishes it. And like here, I sacrifice my student to actually go take them all, compare them against the same condition, the same fine-tuning regime, and and we see it. Maybe at some point somebody will prove me wrong, but we are not quite here yet. So let me just move on and say, okay, we kind of said that method doesn't really work. What can explain to me why we cannot improve? Because this is a real problem. We really, with all the advancement in graph neural network and transformers and others, we do not improve the numbers. And the method that we are kind of looking at, have some hypotheses, we actually look at data attribution methods. Just to remind you what data attribution methods do, they try to explain your accuracy or your mistakes in terms of the specific distributional properties of your training data. So maybe you are more familiar with one called influence functions. So it can, let's say you can take your model, it makes a wrong prediction on some test example, and it can automatically say those are examples that are contributing to this wrong prediction. Or similarly, this is a correct prediction. What are the examples which play the biggest role, which are influencers for this specific prediction? We are using slightly different methods that we developed in the group. And the method is based on the idea that you want to learn to identify the hardest split of your data. Correct? If I give you all the data, can you find me a subset of data that if I put it on the test, my generalization would be the worst? So in this particular case with these Samoyeds, for instance, this kind of generalization would be if you take a Samoyed, a dog, on the snow, and the bear you're going to put on the green. It will be hard because in most of the cases, Samoyeds are on the grass and polar bears are on the snow. Again, this is a goal, to find the hardest split where your accuracy would be low. And why are you going through this exercise? Because typically we want something which actually works better, not to find the hardest split, because this is the extreme case on which you can see your model doesn't generalize. And we developed this method that can actually do it automatically. It minimizes, it's try to find the split going through the ineffective way through the, exponential number of split to find the one in which generalization error is the worst. We did it on this toxicity data set. And what we found was interesting, that this hardest split was a split where majority of the molecules, the ones on which it fails, you can actually see that they all have different targets which are not represented in the training. Remember that this model doesn't look at the target. It just looks at small molecule. It doesn't know what variation of the small molecule to which target does it go. So if your targets are underrepresented, there is no way, however, whatever abstraction you do on molecule itself, you will be unable to generalize. So either you need to completely change your learning paradigm or get the training data which is more consistent with your learning. So the point that I'm trying to say, what we've been doing for quite some time is just trying to take these algorithms that we develop for language or for vision and push them with all the energy we have into chemistry, and we are not improving. So we really need to go back to the fundamentals to see what features our algorithm doesn't capture, and then maybe focus on that rather than trying just to borrow the most recent piece. So another group of algorithms, I want to slightly switch the gears, but this is sort of a success story. This is more generally what we need to do whenever we're thinking about target discovery, and whenever we're really thinking about this interaction. We don't only want to look at small molecule, because we really want to understand what is it doing. For instance, in case of toxicity, with what it interacts to create toxic reaction. Or if you remember the examples that I gave you, you really need to understand how this protein, folds in 3D to connect to it. And I'm showing just one of the cancer drug, neratinib, that's supposed to work with people who have EGFR mutation for lung cancer. And you can see here this 3D model. So the model that we developed in the group are called Boltz. I would talk about it later, but we started with doing re-implementation of AlphaFold 3. And I just want, I don't know, my question, maybe you can raise your hand so that I can decide if I need to talk about it or you already know. How many of you know how AlphaFold 3 works? So we have some and some don't. So let me just highlight computational, because we, again, and here are the 3 students who did the Boltz, not the AlphaFold, but Jeremy, Gabriele, and Sarah. But I just want to kind of highlight some computational ideas that enable to make this, these beautiful models for structuring work. And this is kind of progression of the computational thought. So I'm starting with AlphaFold, and then I'm going to talk about Boltz. Again, whenever what are you trying to do? You're trying to take the protein. Let's just talk about protein. In reality, it can take any biomolecule or pairs of the biomolecules, but let's just make it simple. We're starting from a protein, which is a sequence, and we want to predict the 3D structure. So if you look at the architecture of the model, which is pretty elaborate, there are 2 parts to it. There is a trunk part, which learns how to look at the at the sequence itself, at pairwise connection and multi-sequence alignment, and how to summarize the representation. And this part is called trunk. This is very expensive part. And then there is another part which actually takes this representation and generates from it the 3D structure. So trunk is very heavy because of triangle layers, and the diffusion model, it's actually a light one. Why it is light one? Because you need to do this computation many, many times. So this kind of separation between heavy computations that you do once and the light computations that you can repeat is one of the nicest idea. The second idea, which was again important, if you look at AlphaFold 2, the big jump was how do you model it, moving it from regression to generative modeling. Why this move happened? Because of the intrinsic uncertainty that is part of, of the folding. So what you can see here, that there are 2 possible correct structures when you're looking at the protein and you're looking at the small molecule. So if you're going to model it as a regression, what the model will try to do is to find the solution in the middle, which is incorrect solution. So having the generative model enables you to sample in the space of solutions and identify both of these conformations. And the third piece, which is very interesting interplay according all the chemistry and / biology and machine learning, how much prior knowledge you're going to incorporate and how much biology you're actually going, or chemistry you're actually going to put in. So on one end of the spectrum are models like ESM, a language model, which look at the sequence itself and just encode it in the same way roughly as we would encode any sequence language. And in this case, you really completely oblivious. You don't incorporate any information about structure. You just operate at the sequence. You embed it into a vector and you operate in this space. On the other end of the spectrum are early AlphaFolds like AlphaFold 2, where you start incorporating more constraints. So you explicitly incorporate on the model rotation invariance, translation invariance, and other things. But the problem with this kind of direct forcing the model to have the right features comes with poor optimization dynamics, poor scalability. And what was really nice in AlphaFold 3, that with this specific with this architecture actually enabled you to learn all this type of knowledge directly from data through the augmentation. So you are learning all these features without forcing the model to do. You make it aware of the constraints, but it actually learns it on its own. And again, we started with this Boltz just to reproduce AlphaFold 3. The students who started doing it, I thought they are crazy and they would never do it because we don't have engineering team at MIT, but they happen to be super crafty and they proved me wrong. So in this case, we started by doing it. And the biggest advantage is that AlphaFold 3, those of you who use it, it's you can run it, but the training code is not available. So if you want to fine-tune it, a particular class of biomolecules, you could not do it. You can actually do it with Boltz. And another big addition that happened with with Boltz is that we can now predict affinity. So we can not only predict the pose, but we can predict how strongly the molecules interact. And it's really very commonly used. And you can see it's quite used model. I just want to show you one example. It's like really recent example of how this model is used. And it comes from our collaborator, Justin Gaynor, who is a professor. He's a professor, I think, but he's a doctor at MGH. And the question that he asked is, it's kind of a question that every oncologist faces when you have targeted therapies. Targeted therapies for cancer are the ones when, again, you have mutation in some protein, let's say EGFR, and there are drugs that just work, kind of fix the mutation, like what we see at the beginning of the talk. The point is that today, first of all, you can have many different mutations in the EGFR, and you also have a problem. There are many possible drugs. Every year, pharma industry continues to produce new drugs for this mutation, and people don't know which drug you should give to a given patient. And there was this paper, I think from Nature a few years back, that shows that if you can crystallize the specific mutation and the small molecule, you can actually reason about the efficacy, because each of these drugs goes to a different pockets of this protein. So if you can model their connection, you can say what is likely to work. And what we've demonstrated that if you're going to be doing it with Boltz, if you're just going to take the mutation and you're going to take the specific EGFR mutation and the molecule, you can find the one which fits the patient better. Now, the thing that was published in Nature, you actually need to crystallize, which is a very expensive process. You would never be able to do it for any mutation of the patient. This stuff you can pretty much run in the lifetime. I cannot tell you that it's already run at MGH, but this is really promising to show that even besides the drug discovery, these tools really work very well. Another example, this is my student Hannes's work, is to generate the binders. Yes, yes. You're talking about this slide? Yeah. So in this case, we're just taking the small molecule and the EGFR, the sequence of this variant runs through Boltz, and we take Boltz probability, the probability of binding. We just use one score. This is like really a new work, and we started with EGFR. Now we look at some other targeted therapy, and maybe we can do even better if you add, really to look at the interface and look at other things. This is like the simplest thing, but we already see, even with that, you have a signal. So we saw it both on cell lines and on patient data. So we now need just to do it in a elaborate way. So here I just want to show you another tool that came out in November. This is called Boltz. When I showed you Vertex drug, I showed you identify the place where the mutant misbehaves and you want to fix it. So Boltz, you can give to it which protein you want to have a binder for and where to bind and it, or not. It can find your location. So here you see a protein which is implicated, a target which is implicated in many cancer. Telomerase, we don't have a good binding, and this is a small peptide that the tool generates. And now there are a lot of Bindcraft and other tools, and you can say, why would I want to use Boltz if there are so many great alternatives? There are two features that I think are attractive about this tool. One, it can be generalized across modality. Your protein can be in any form. It can be small molecule, it can be protein, etc. And you also can decide in which modality to generate the binder. And here you can see some examples across modalities. And finally, it also generalizes in the space of targets. That, we don't want to generate more binders to the same protein, that we already know how to do it. We really want to move into the space of undruggable targets. And we've demonstrated through multiple experiments that Boltz actually can do it. And here you can see some examples in a wide range of therapeutic modalities for 26 targets that we validated with multiple collaborators. And we continue doing the validation. So this is some of the molecular tools that I wanted to show you. And I just very briefly, what time is it now? Yeah, let me just very briefly talk to you a bit about biology, because I was very solidly in the chemistry space.
Excuse me,
yeah.
After you generate the binder, do you assess the binding score with Boltz 2?
No, so the way we do it, Boltz 2, whenever we do it, like there is a preprint which describes it, of course, in multiple details. You do use. Actually, what's interesting about Boltz, that architecture is such that it's kind of reversible. It can both use for structuring and for generation. So it actually models, in order for it to generate this refined binder, it needs to reason about 3D geometry of of whatever it generates. But afterwards, after we generate it, there is this filtering pipeline that we, for instance, if it is a protein and small molecule, we can run Boltz 2 to check if it is the right affinity. So there is another filtering step, which is a bit ugly, but unfortunately, most of the model have this pre-filtering space where you use these extra tools to limit the most promising candidates. And I should say that after we give to collaborators these candidates, we typically give them, depending on their budget, 20 or 50 candidates, they go and do their experiments. So what I showed you, it's not, we generate millions of things. Those are the ones that actually been validated by the experimentalists in the lab. Yeah. So, and let me just talk a tiny bit about biology piece, correct? So I want to show you this paper, and I'm carrying this paper with me. It's an old paper from New England Journal of Medicine, and it was the first paper that I read in my life from New England Journal. And this paper asks a question. Women who were premenopausal, who have breast cancer, should, in addition to their treatment, should they get another drug called Lupron for variant suppression? This drug has plenty of side effects, but people may still want to take it if it really saves their lives. So what you see here, and I remember this paper by heart with tables. It's a very old paper. You can see the red line and the black line show to you the improvement in survival. So there is barely any effect, correct? Barely any effect. And here what you can see on the right, it's an impact for different subgroup. It's a very coarse description, based on age, based on tumor type and so on. Really, you cannot make any sense of these numbers. You have no clue should you be taking it based on this data. Maybe there is some other data, but not based on this data. Now, if you are looking at the papers that are produced in biology, where people are really trying to understand why somebody is a responder, what is the biology of cancer, they never work on the data from clinical trials. They work on a very separate data. They do single cell, they do spatial transcriptomic, the stuff that you would never do in clinical care. And the question that we've been asking for the last year is, can you take normal data that people actually collect in clinic, not something very special, and understand the biology of cancer? Can you get some insight? Can you understand who is going to be responder? And I was very fortunate with MIT group and MGH group, which is technically Harvard, to participate in this big program of ARPA-H, which is called ADAPT, that does something really fascinating. It tries to use AI to find the best treatment for metastatic patients. As you know, patients who are metastatic have very not bright outcomes. And, after they fail the first line of treatment, then, they select the second line of treatment. And for most of them, their lifespan is relatively short. And the hope is that if you can very accurately select what's likely to work for the patient, you can prolong their life. And what you want to do, you can say, let me collect from patients, let's say, their RNA. I know what is a drug and see for how long this patient is going to live without getting recurrence. The way, there are I realize there are two camps today that think about this question from very different perspective, and they typically never interact. So the first camp is my camp. This is a camp of people who really do Boltz and do molecular and atom models and stuff like this. This is what we do. And then there is another camp, which the way they're thinking about this question in really thinking about cells. They don't really look at the kind of atomic interaction. They just look at cell and say, okay, I have a cell. For instance, I can look at RNA-seq of the cell, correct? I can measure it. I look at the RNA-seq of the cell. I have my perturbation, let's say a small molecule, and then something happened. I do perturb my cell, and then there is a new state. So you can say that I am actually trying to model. There is an input, we put something into cell, and then the change happens. And what I'm trying to do, I'm trying to go from the original state to some state that I consider to be disease-free state. So, and typically this model is kind of black box model. You have the data, it's perturbation data. For those of you who are familiar, there are a number of models of this sort, like state and others, that you take a cell, you take a perturbation, then you see what happened in the end. Doesn't really work very well. This is another example of travesties that's going on. Now, I should say that this data set continued to be released. Even the last month there was one released where people do these huge perturbation data sets and try to demonstrate that now, because they can represent cell with some amazing foundation model, the miracle happens. And now you would know what will happen, correct? For those of you who, I'm just showing you some examples of data sets. Like you can see, it's pretty large data sets which can be used to learn how to represent cell. So if we're just going to stick to RNA-seq, you can imagine that RNA-seq, it's a measurement of, it's an array of 20,000 genes. And the question, how do we represent it? Now, in the old times, before the deep learning came into our life, people use more biologically, bioinformatic things. For instance, they would use gene sets. Gene sets are some subset that was through the very deep biology was implicated in, let's say, are relevant to cancer biology. There is something else where you don't even need to understand biology, which is called highly variable genes. You take your population and you identify which ones of 20,000 genes change the most across the population. And you can just use very simple dimensionality reduction. What you would assume, correct, that this type of trivial, simplistic representation would never work better than the fancy trained on millions of data points foundation model. So let's just look at the data. And again, there are lots of architectures, what people do. You can start already feeling something is askew after you look at this paper from Nature Medicine that came out, I think, maybe last year, correct, or 2 years ago? No, last year, that demonstrated that if you just take an array itself without doing any processing, without doing any embedding, without doing any foundation model, it already works better than the foundation model. So you can say, okay, and then people from some other places say, they don't measure it correctly, whatever. I feel so angry about this topic because when we started working on ARPA-H, I burned 3 months of my students' time and saying, we need to make this foundation model work. People are publishing papers, people from good places, something is wrong with us. So after 3 months, I am convinced it's not us who is at fault. And what we've done, actually, after 3 months of this failure, I just decided to investigate more broadly. Can you use these foundation models for cells across the task? And typical task for predicting in cancer are two relevant tasks. One is predicting disease-free survival, for how long something will live, and the second one is how it's going to respond to medicine. And what you can see here, those are different classes of models. You can see all the most recent single-cell, bulk RNA models, whatever. The best models here are the one in green. What are the ones in green? Here we're looking gene sets that biologists combine. Here we're looking at highly variable genes. Here we are looking at raw expression. So you can see in this case the trivial, the most stupid model works better than foundation models that continue to be published every year. Here I'm just showing you the result. This is actually interesting. This is a model from Harvard Compass, very sophisticated foundational model. No comparison, no, in the data set that was published in the paper, no comparison to using the most trivial baseline. This is kind of really upsetting to me, what's going on in the field where we burn time without even understanding why are we applying these sophisticated methods that don't really work. And I would just very briefly tell you, what we did in this case to predict response. This is a work of my student Serena Ko. What we've done here, we kind of started with, we said, okay, these gene sets seem to be promising. This is the pathways that people identify. And these pathways, it's like a vocabulary of them that on the biochemical level explain to us what this molecule actually do to cells. So there is a limited vocabulary that is well identified, and we focus on learning how to represent the embeddings of these pathways as vectors, and then we represent the molecule itself as a weighted combination of each pathways, because depending on your drug, some pathways are more important to be activated than others. So you just take RNA-seq, you map it into these gene sets, and you learn the embedding, and then you do the weighted combination and predict response. So we've demonstrated that it works pretty well. We also discovered some new pathways, or pathways that are really critical to a particular drug for the responders, and now you can just take this pathway and validate them in the lab, and that's what we are doing. But in terms of response, you're already getting much better numbers. I have some other stuff, but I think it's a good point. I don't want to run over time. But what I demonstrated to you, that this view of taking stuff from other field and bringing it to biochemistry is very non-productive approach. And if we want to improve, it's really understanding what doesn't work and also being honest to ourselves and actually take the simple baseline and see, are you adding something with your sophistication or not? And the dilemmas we've seen already in these biomolecular models, how much do you really encode the biology and chemistry? How much you are learning kind of freestyle? It's an open question. The huge advancement in biomolecular model really show this trajectory and demonstrated that being creative about modeling really yielded some important results. And I hope that this creativity is going to be spreading to other areas of biomolecular modeling. So thank you very much.